Only one way to find out. I'll get back.
Good morning, everybody. Can you hear me okay? Am I coming through the speaker?
Did that work or no?
Yes. Okay. Welcome to day 2 of IHO. Any questions so far? Everything going okay? All right, so the topic for the next 2 hours, we're going to touch on some general features of blood and some of the chemical composition of blood and some of the physical properties of blood, and that's kind of one side of it, and then another side of it will be some of the metabolic processes that are happening within red blood cells. Okay, so some stuff is going to be happening outside of the cells, and then some stuff is happening inside of the cells. So we're gonna, it's a little bit of a hodgepodge today. I think that the objectives are pretty straightforward, I guess, with the exception of this one, hypoalbuminemia. That's referring to decreased serum albumin, and I don't think I use that word for it, but protein malnutrition or kwashiorkor are two things that would result in that. Okay, please sign your tablet or phone or other mobile device or what have you into the personal response system session ID as indicated on the screen there. I'll have start out as usual with some clicker slides, and then I've got a few kind of scattered throughout, and then if I get the timing right, we'll conclude with a clinical case. All right, so let's review some foundation stuff. Okay, majority of people picked anaerobic glycolysis. What about glyceraldehyde 3-phosphate dehydrogenase? Doesn't that generate NADH in glycolysis? It does, but anaerobic glycolysis, remember the product isn't pyruvate, it's lactate. Okay, so that NADH that's made by the glyceraldehyde 3-phosphate dehydrogenase, that's going to get oxidized by lactate dehydrogenase. So there is reduced NADH generated, but it's oxidized as part of the process. So maybe no net reduced NADH would be a better way to explain this. I'll elaborate on this concept later. Okay, this one I'm sure is tattooed in your central nervous system from foundations. All right, good. PFK1, very good. So you know what's coming next, right? Remember, PFK1 has that complex allosteric regulation that makes it sensitive to intracellular energy conditions, availability of substrates, and there's also a endocrine regulation as well. So it's going to be activated by things that indicate abundant substrates or low energy, and it's going to be inactivated by things that indicate abundant energy. Okay, so fructose 2,6-bisphosphate, that's the product of phosphofructokinase 2. That's one of the things that activates PFK1, and adenosine monophosphate is another. And then it's inhibited by both citrate and ATP. So those are indicators of abundant carbohydrate and high energy, respectively. And remember that all cells have this ATP-dependent pump that maintains this relationship between these two cations, right? If that helps you remember it. That ATP something something exchanger. Okay. Okay, so extracellularly we have high concentrations of sodium, and then potassium is concentrated within cells, or at high concentrations within cells. Okay, 100%. Dynamite. Great job. Okay, so let's start with a little didactic lecture, and then keep those clicker response devices handy, because I'll have some more questions coming up soon. Okay, so composition of blood. Blood is what's circulating around within us, of course, and we want to maintain it in that fashion. So we'll talk about coagulation later in the course, towards the end of the course, and that's kind of how we maintain blood being inside us instead of outside of us, so we'll kind of punt on that topic. So if you look at that blood, like blood is the kind of all-encompassing term for both the non-cellular and the cellular stuff that is circulating through our veins and arteries. So if we take a centrifuge and separate out the cellular fraction from the non-cellular fraction, then we get plasma. Okay, so plasma is the non-cellular portion of blood. And then if we let that plasma coagulate, then we call it serum. So they're kind of, blood, plasma, and serum, and they're kind of the same thing, but they're also subtly distinct. So make sure that you have those definitions down. Now, one measure of whether a patient has too many red blood cells or too few red blood cells or just enough red blood cells is the hematocrit. And the hematocrit is the fractional volume of red blood cells within a blood sample. Okay, so you can get it by spinning the red blood cells out, as shown here, and just measuring the height of the red part of it. Or if you're trusting, you can just take the hemoglobin number and multiply it by 3, right? That's always a pretty good estimate for what your hematocrit will be. But they frequently measure both of them anyways, even though they go along very well with one another. Okay, let's see. Oh yeah, the buffy coat here. This is the cellular portion of your blood that is not red blood cells. So the red blood cells are more dense than the other cell types, and they're going to be at the bottom of the centrifuge, and then your platelets and white blood cells and what have you are going to be a layer on top of the red blood cells. Okay, so looking at that plasma or / serum and looking at the ionic composition of it, or the electrolyte composition of it. So sodium, that's going to be the major cation in plasma. And then chloride and bicarbonate, these are the two major anions. I'm going to talk a little bit about chloride and its relationship with bicarbonate and how that buffers pH in plasma. You're going to get a lot more of this in the fall of your second year in the CRAB course, the cardiovascular renal respiratory acid based stuff. So talk a little bit about electrolytes and pH, but you're going to get a much deeper discussion of that in year 2. So, some topics I will skim over. Okay, so that topic of the salt composition or the salt concentrations within blood, let's just kind of punt on that, and we'll nibble off little bits of that here, and then talk more about it in year 2. Okay, so beyond the salts in our blood, blood is also rich in proteins, and I should say plasma and serum are rich in proteins. The cells in our blood, those are very concentrated in proteins. But if we just consider the acellular fraction of blood, that has very high concentration of proteins, like in the milligrams per deciliter range. And those proteins are diverse and discrete. It's not like a mishmash of all different random proteins. There are specific proteins that have specific functions, of course, as you can imagine. And if you look at the concentration ranges of the different proteins, the concentration ranges vary over vast orders of magnitude. Okay, so some proteins like albumin, which is the most abundant protein in the blood, that's in that grams per deciliter, very high concentration range. And then proteins that act like hormones, like gastrin or insulin or something like that, that's going to be present in, I don't know, picograms per milliliter, very small, very low concentrations. So any one particular protein in the blood is going to be, it might be there in vast amounts, or it might be there in tiny amounts, but hopefully it's in the appropriate amount for whatever its function is, right? Okay, so if we take a sample of serum and we precipitate out the proteins and then separate them electrophoretically based on their charge and their mass, we can visualize the diverse sizes and the diverse abundances of the proteins. And that's shown in that, let's see, I'll use my marker here. And that's shown in this strip over here, where we have, the larger proteins are up on the top here, and then they're arrayed with the smaller proteins migrating faster in the gel. And then the intensity of the band, that is proportional to the abundance of that protein in the sample. Okay, so a couple things that I'll point out here. So albumin, this is by far the most abundant protein in the blood, and it has diverse functions, and I'm going to talk about all of them one by one shortly. So albumin is made by the liver, and it is it's kind of like the styrofoam packing peanuts of the blood. Okay, so it's there just to bulk up the blood with protein, and that has a lot of consequences. Okay, so Dr. Diebel talked about immunoglobulins just recently, right? So a lot of the other abundant proteins in the blood are immunoglobulins, and I'm just going to leave it at that because I think he's had a good discussion of it. Now, this diagram is showing a sample of plasma, right? So what would be the difference between the protein concentration in plasma versus serum? Well, what's the difference? In serum, it's clotted or coagulated, right? So you're going to get coagulation factors are going to be precipitating out. Now, a lot of those coagulation factors are not abundant enough to be shown on a electrophoretic graph like this. You'd need a more sensitive detection technique. However, there's one, fibrinogen, which is this band right here. And this is the major end result of the coagulation cascade, as we'll see in a few weeks. So if you just let that sample of plasma sit, or if you add some positive charge to it to accelerate the coagulation process, you're going to lose that fibrinogen band. So that fibrinogen makes a polymer, and that polymer is, a solid and precipitates out of the solution, and then it's no longer soluble in the blood. So the difference between plasma and serum in terms of those very abundant proteins, it's going to be the absence of fibrinogen that is going to define the two. Okay. All the other proteins, that aren't involved in the coagulation cascade, they're really unchanged between serum and plasma. Okay. All right, so should be a pretty easy question for you. I guess I maybe should have saved this for the end, huh? Okay, super. So we'll have a lot more to say about coagulation. I'll do the biochemistry of it. There's a pathologist who will discuss disorders of coagulation, and Dr. Regal will talk about pharmacology of coagulation. But that's all going to happen like in the last 2 weeks of the course. Okay, so just put a pin in that. Okay, another question here. Well, we did discuss this, or maybe a little bit. Okay, good. We must have talked about this. So as fatty acids, free fatty acids bound to albumin. So just, shorthand to remember it, fat is going to adipose tissue in a lipoprotein particle, like a chylomicron or a VLDL particle, and fat comes from adipose tissue as free fatty acids, right? But those free fatty acids are not soluble enough to be a free fatty acid floating around in the blood. They have to be associated with a hydrophobic protein like albumin that can allow them to be transported. Okay. All right, one more question, and then we'll talk more about albumin. Have you had a lot of cancer pharmacology? No? Okay, get ready. It's gonna come fast and furious in a couple of weeks. Okay, so paclitaxel and docetaxel, these are two drugs in the taxane class, and they act on rapidly dividing cells in M phase, and they prevent the depolymerization of the microtubules that are pulling the chromosomes apart during M phase. So basically the cell can make microtubules, but it can never shorten them to pull the what do you call them? Chromosomes, let's just say, apart. So you get the cells dying by a mitotic catastrophe. Okay, so that's the mechanism of action. Okay, and I'm going to talk about a lot of classic cancer therapies and some newer emerging cancer therapies in those 2 weeks of the O portion of the IHO course. Okay, so That's coming up. Okay, so what I really want to talk about is albumin. Albumin is the most abundant protein in our blood. It far exceeds the concentration of the next most abundant protein in the blood. Okay, so albumin is synthesized by the liver, and albumin is exported from the liver and it enters into the blood circulation. It can then exit the blood circulation by a transendothelial cell transport and enter into the interstitial fluid in tissues. So there is a force that is propelling the albumin from the blood into the tissue and then into the lymphatic circulation. And I'll discuss the nature of those forces shortly. And this is really driving that lymphatic drainage of the tissue, and it allows the lymph nodes to interrogate our body and our different tissues and mount an immune response when it's appropriate. Okay, so albumin has this circulation, but really the most albumin in our bodies is present in this interstitial fluid here. Have you ever cooked salmon and then seen that white little blobby stuff coming out of it when it's cooked? Okay, so that's albumin, or most of that is albumin. That's being denatured and then aggregating and kind of coming up out of the interstitial fluid. Okay, so what's the function of albumin? Albumin has diverse functions. One is it's important for transport. It is critical for maintaining oncotic pressure within our blood, and that is the driver of that transport of fluid from blood to tissues to lymphatic vessels. Albumin also has functions as an antioxidant. Okay, so as far as the transport functions of albumin, albumin has several different hydrophobic pockets. So there are regions on the surface of the protein, but they're made up of, or the amino acids that are making that surface have hydrophobic properties, like valine and isoleucine, etc. So this allows the albumin to bind other things that are hydrophobic, like free fatty acids. So that's shown here. This is the carboxylic acid end of a free fatty acid, and it kind of can wiggle into this by interacting with those hydrophobic amino acids that make that pocket. So there's one shown here. There's another site up here. I think there's another little hint of one there. I think there's like 7 different sites where albumin can bind to free fatty acids, and those are critical for that transport of the free fatty acids from adipose tissue to the liver and to other tissues that like muscle that would oxidize the fatty acids as a metabolic fuel. So in addition to fatty acids, there are many other endogenous substances like bilirubin that are transported in association with albumin, as well as many different drugs. So a lot of drugs are too hydrophobic to be easily soluble in the blood, but they can be transported because of that ability of albumin to bind to hydrophobic substances. Okay. Now that binding and delivery of things by albumin, that's not just purely within the circulation. It's not just delivering it to an endothelial cell, but albumin has this property of being amenable to transendothelial transport through interaction with a receptor that's present on endothelial cells. That receptor is called GP60 or albondin, and this feature allows the albumin to transit across an endothelial cell and then concentrate in that interstitial area. So this is showing a capillary with an endothelial barrier and then tissue. This is an electron micrograph. And the black dots are albumin particles that are labeled with gold. So they're really very electrodense and show up bright on the electron micrograph. So you can see that there's not a lot of the albumin out here in the lumen, right? So it's concentrating at the surface. So that's indicating receptor binding, right? So it's binding to that GP60 receptor. Then you can see an invagination in the endothelial cell here. So that's receptor-mediated endocytosis. And then here's a little vesicle that's transporting the albumin across the endothelial cell and into the interstitial or the perivascular or interstitial area here. So just kind of nice demonstration of that feature of albumin of crossing that endothelial barrier and delivering its contents to the tissue. So this is important for albumin's normal function within our body, and this feature has been exploited therapeutically to deliver cytotoxic drugs to cancer cells. So that endothelial or circular or the blood vessel barrier is something that you have to overcome pharmacologically in order to deliver a drug to a cancer cell. And albumin is one method, one FDA-approved method that can do that. So there is a molecule or a drug called Abraxane, which is a big glump of many albumin molecules all polymerized together and then linked to the paclitaxel cytotoxic drug that stabilizes the microtubules, as we discussed earlier. So the idea here is that the conjugating the paclitaxel to the albumin will increase the concentration of that drug into the tissue where it belongs rather than in the circulation where it's not really doing much for a breast cancer patient. And have we seen forest plots before, looking at clinical trial data? Seen this? Okay, so it's basically, if you're comparing a placebo versus a drug, you are looking for, depending on what your outcome is, if it's on, you have a one line here, and if the outcome favors the placebo, it's on this side, and if it favors the investigational drug, it's on this side. And then you can do your overall results, or you can stratify different subpopulations within your experimental cohort. Now, in this case, this was a large clinical trial that was comparing albumin-bound paclitaxel with just regular paclitaxel that would be soluble and delivered without albumin. And this was people with breast cancer that had disease progression while on other therapies. And you can see that there's a pretty strong effect overall that the albumin-bound paclitaxel was an advantage. Now, these big lines here, or these are referred to as whiskers often, if they're overlapping that number one, that kind of no effect, then that's not a statistically significant result. Okay, so the overall result here strongly favors Abraxane or the albumin conjugated paclitaxel versus the solvent-bound paclitaxel, but it's not for all groups. So we know let's see, I guess have to remember that this year for you, we removed a breast cancer module that was in fundamental or in the foundations course. So you haven't really talked about breast cancer at all. And the breast cancer subtypes? No, not at all. Okay. Okay, so we'll maybe touch on that at some point, hopefully. So the majority of breast cancers are driven by estrogen and progesterone signaling, right? So those would be the luminal A or luminal B subtype. So that's the majority of cases, and the majority of deaths are with those subtypes. If you don't have hormone receptor, and if you don't have another driver of breast cancer cell proliferation, the HER2 growth factor receptor, we refer to those cancer cells or that subtype of breast cancer as triple negative or basal subtype, it's often called. And that is a rare subtype of breast cancer relative to the hormone receptor positive subtypes. However, it strikes women at a younger age and it tends to be more aggressive. And because it doesn't have the HER2 and it doesn't have the estrogen receptor, it doesn't have two major targets of therapy that you could otherwise treat the cancer with. So that's like a subtype of breast cancer that is, quite undesirable. Not that anybody wants to get breast cancer. Okay, so long story, but that, let's see, where is it? The HER2 minus hormone receptor minus subtype, that had the biggest benefits of the albumin conjugated paclitaxel relative to the non-albumin conjugated. Okay, so long story, but if you have that type of breast cancer, it's pretty significant. Okay, questions about this? So this is called a forest plot, and it's a common way to view clinical trial data. All right, should we do a clicker question or two? I guess, I'm sorry, just one. Okay, so beriberi is thiamine deficiency, and pellagra is niacin deficiency, and pernicious anemia is vitamin B12 deficiency, and scurvy is ascorbate deficiency, right? Vitamin C deficiency. So that leaves kwashiorkor as protein malnutrition, and yeah, majority of you have that. Okay, what's a prominent clinical feature of kwashiorkor? Yeah?
Like children's stomachs really expanded.
Yeah, that's right. Yeah, so a prominent swollen belly from edema. That's kind of a classic hallmark symptom of kwashiorkor, and especially in children. Okay, so that edema that results from protein malnutrition, that's related to albumin's function as providing oncotic pressure within our blood. Okay, so let's talk about that oncotic pressure and what that means. Now, we talked about when we were introducing albumin, that flow of liquid from a blood vessel into the interstitial space and then into the lymphatic circulation, and then that ultimately will rejoin into the blood circulation. So if you look at any particular capillary, you're going to have the arterial end of the capillary and then the venous end of the capillary, right? It's because the blood goes from one direction to the other. Okay, so that's what's shown here. And blood is flowing from one direction to another, right? So that means there must be a pressure gradient. The pressure that your heart is generating, that's going to be higher on this end than on this end, right? And that allows the blood to flow from high pressure to low pressure. Okay, so that hydrostatic pressure that's generated by your heart pumping, that is one or two of the Starling forces that determine how much fluid is going to be leaving the blood vessel. So the Starling forces are the hydrostatic force at the arterial side and the hydrostatic force at the venous end of the capillary are two of the forces. And then the other two forces are the oncotic pressure at either end of, at the venous end and the arterial end of the capillary. So oncotic pressure, this is like a osmotic pressure that is derived from the colloid albumin. Okay, excuse me. More precisely, it's pressure that's derived from the concentration of all of the proteins within our blood. But because albumin is by far the most abundant protein, albumin is the major contributor to the oncotic pressure within our blood. Okay, so in a normal situation, in a healthy person, that oncotic pressure is going to be remaining fairly constant throughout the capillary, but at the arterial end, the hydrostatic pressure is going to be exceeding the oncotic pressure. So you're going to get liquid leaving the blood vessel and entering into the interstitial fluid. And then at the venous side, the hydrostatic pressure is lower than the oncotic pressure, so you get fluid returning back into the blood vessel. Okay, so on net, you get a little bit of fluid going kind of one way into the lymphatics, and then that allows the tissue drainage. But we get a significant amount of fluid returning back into the blood vessel at the venous side of the arterial. Okay, and that's in a healthy person with adequate protein in their diet and adequate albumin concentration. Okay, so that's shown here. Normally on the arterial side, you get a loss of fluid out of the vasculature because the hydrostatic pressure is exceeding the oncotic pressure. And then on the venous side, you get a refilling or a draining of the interstitial fluid back into the blood vessel. Okay. Now, yes.
I just have like a clarifying question. So I was like thinking of it as like a garden hose, and if you have high osmotic pressure on your venous end, wouldn't it want to go like out? Like if you have high pressure in your capillary on that side, is that what that's saying?
Well, it's that, I guess the hydrostatic pressure is pushing the liquid out on the arterial side. And then that hydrostatic pressure is less on the venous side, right? So
it's more of like outside in.
Yes.
Okay.
Yes. I think that's correct. Did you have a?
Is it just because of like gradient, concentration gradient of albumin from the lymph to the like venous?
It's an osmotic effect, right? So oncotic pressure is a type of osmotic pressure. Yeah. So, and that's probably the easiest way to think of it. Any other thoughts here? Okay, so it might be easiest to illustrate what's happening if you think about the condition of kwashiorkor, where you don't have sufficient albumin in the blood. Okay, if you don't have sufficient albumin in the blood, then the oncotic pressure is going to be reduced all along this gradient, or all along the capillary. And the consequence is there your hydrostatic pressure is going to kind of outweigh the oncotic pressure at both sides, right? So you're going to get loss of the fluid on the arterial side, and then you're not going to get enough replenishing of it or refilling of it on the venous side. So that results in the accumulation of fluid in that interstitial, in the tissue, and that results in the edema and that swollen belly. That's kind of the hallmark symptom or hallmark physical exam finding of pediatric kwashiorkor. Okay, is this all okay with everyone? Yeah.
I guess just one last question. question. If albumin, if a lot of it is not in the lumen but in the interstitial fluid, how come it doesn't want to pull the water out? Like, I don't understand. I guess I would assume that the albumin being inside the lumen is what's causing that oncotic pressure.
Yeah.
But a lot of it is also outside the lumen, so how does that happen?
I think, like, if you looked at all of the albumin in our body, the majority of it would be in that interstitial space. But if you looked at the concentration, milligram per unit of volume or whatever, it's higher in the blood than in the interstitial.
Okay, thank you.
Yeah, is that okay? Is that
Makes sense.
okay? Okay, so take a look at this on your own, and if you have questions about it, we can discuss this more on Thursday. But I think maybe just kind of, take a look on your own and try to chew on it a little bit. Okay, so what's the final function of albumin? Albumin has a role as an antioxidant. It has a free thiol group that is derived by a cysteine amino acid. So cysteine has that free sulfur atom, and it's shown here in the reduced form. Now we know that sulfur from a cysteine, it can be oxidized by making a disulfide bond with another sulfur on a cysteine, and that's the case with glutathione and with a lot of what are those sulfur-containing redox proteins called? Can't remember. Okay, anyway. So that's one way that that a thiol could be oxidized is by joining with another thiol. But you can also get a more, more familiar to you if you're thinking about carbon oxidation, where you can get the sequential replacement of the sulfur hydrogen bonds with sulfur oxygen bonds. And you can get, this is kind of like a carboxylic acid with sulfur, if you will. And then this is I'm not sure what, so this is German here, and I don't know what that is. Anyway, this is how that that sulfhydryl group on the albumin can get oxidized, and that allows it to act as a single electron sink and just as a non-enzymatic antioxidant in our blood. Okay. All right, so we've talked a lot about albumin. So which of these is not a function of albumin? And honestly, albumin does so much in the body, maybe you could make a argument that because it's carrying metformin around or something, that it would promote glucose homeostasis, but I think this is the answer that I had in mind. Okay. All right, very good. Okay, so let's consider our mechanism of edema in kwashiorkor. Okay, very good. So hydrostatic pressure exceeds oncotic pressure in capillaries. That's the best way to remember that. Why do we get hypoalbuminemia in kwashiorkor? Did we talk about that? Why would you think?
Not enough in diet.
Yeah. So you need amino acids to synthesize albumin in the liver, right? And if you don't have protein in your diet, your liver doesn't have the substrates to make albumin. Okay, so it's that simple. And if you're in a starvation situation, your body, your liver is going to be prioritizing other stuff than making albumin. Okay, questions? Yes.
So you were talking about the antioxidant effects of albumin. Is there an enzyme that like converts it back after it's been oxidized, like glutathione?
I don't think that there is. I think that's like a one-way trip.
Just kind of trashed.
Yep.
Okay, cool.
Yeah, that's a great question though, and I'm gonna look that up when I get back to the office. Because after I said that, I just realized that I said that because I haven't heard of one, not because I know that there isn't one. So okay, good question. Any other questions? Okay, so let's segue to another topic, and that is the regulation of blood pH. And for this, we're going to have to get into some of the processes that occur inside of cells. Okay. All right, so the maintenance of blood pH, I guess, why do we have to maintain blood pH? Because we are always producing carbon dioxide through respiration, right? The TCA cycle is making a continuous supply of carbon dioxide, and if that built up, that would acidify our blood, and none of our enzymes would function, and we'd die, right? So we have to have a way to regulate the pH both of our extracellular fluid and to our intracellular fluid. The pHs of both of those compartments have to be regulated, and we have to get a way for this carbon dioxide that's produced through respiration, that has to leave somehow, right? Because we're not plants, and we can't take that carbon dioxide and use it to synthesize new biomass, right? So we have to get rid of it, and how we get rid of it is we breathe it off. Okay, so when carbon dioxide is generated in tissues, that is can enter into the blood and be taken up by a red blood cell, and that can be a substrate for the carbonic anhydrase reaction. Carbonic anhydrase makes the carbonic acid, and the carbonic acid then is in equilibrium with bicarbonate through the dissociation of one proton. So this carbonic acid bicarbonate buffer, this is very important for extracellular buffering of pH. So, and this is a cartoon that shows the basic relationship between a respiring cell and a red blood cell. This is showing as an example a hepatocyte, and that's for a reason, but really as far as this upper part here, this taking a diverse dietary fuel and converting it into acetyl-CoA and then oxidizing it to carbon dioxide through the TCA cycle. This could be a muscle cell or a brain cell or any other cell. Okay, so just general respiration is going to be generating carbon dioxide. This carbon dioxide can diffuse out of the cell that produced it, be taken up by a red blood cell, and then that carbonic anhydrase reaction makes carbonic acid. The carbonic acid then dissociates into bicarbonate. So that would have the effect of acidifying the intracellular environment of the red blood cell. Intracellularly, the red blood cell pH is buffered through, well, in any cell, the intracellular pH is buffered by a phosphate system, phosphate buffering system, and then through protein buffering. In the case of a red blood cell, the most abundant protein in a red blood cell is hemoglobin, and that either unprotonated or protonated hemoglobin and the equilibrium between these two, that's one of the important intracellular buffering systems. And then we also have the phosphate buffering system that can also accommodate changes in the proton concentration intracellularly. So that's, again, this side of the intracellular buffering is similar with the red blood cell as it is with any other cell in the body. So we've got, let's see, the proteins buffering here, and that's, not going to be hemoglobin. In another cell, it's going to be other proteins, and then the phosphate buffering system. Okay, so what about the fate then of this bicarbonate? So bicarbonate is the major extracellular pH buffer. And why do we need to buffer the extracellular environment? Well, cells make organic acids, as shown here with the hepatocyte is doing fatty acid beta oxidation and then generating ketone bodies, right? So that's going to be an acidification process. Likewise, the red blood cell, not shown here, but the red blood cell is going to be making lactic acid, and that's going to dissociate into lactate and a proton, and that will have the effect of acidifying the extracellular environment. So the bicarbonate, this leaves the red blood cell through a chloride bicarbonate exchanger, and the bicarbonate then can act as a pH buffer by being in equilibrium with the carbonic acid extracellularly. Okay, so looking at the clock here, why don't we take a break, and we'll elaborate on this topic at 11:01. Sounds good? All right.
Yay.